All right, welcome back from that little hiatus. Hopefully, you're all caught up with the test and everything, and we're able to get chapter 16 done out of the way, and that you're ready to start chapter 17. So, this chapter is going to be about nucleic acid. We're going to start the first lesson talking about nucleotides and nucleosides. So, our first focus is differentiating between a nucleotide and a nucleoside, and you'll see that those ultimately end up making up nucleic acids, which, by the way, you should be more familiar with, commonly known as DNA. So this is the fourth macromolecule we were discussing. It's basically DNA, but there's also RNA as well, and so I'm saying if you hear the word nucleic acid, you might have no clue what that means, and most people don't, but it refers to DNA. And then that will wrap up the four major molecules of life. Isn't it kind of interesting that there are essentially only four major molecules within us that make us up? I mean, there's other stuff too. There's water and inorganic compounds and air and other living bacteria within us that make us up. But essentially, we're made up of four different types of macromolecules: sugars, fats, proteins, and nucleic acids. Now, this is what's really interesting, and I'm just going to say this real quick before we dive right in. DNA, or nucleic acids, eventually code for proteins. That's all they do. And so, which one's we should have a debate on which macromolecule is the most important. DNA, proteins, carbs, or fats. They all have their own purpose, and DNA's purpose is to make proteins, is to literally make one of the other macromolecules. So we really only have three macromolecules that are important: proteins, carbs, and fats. But DNA is required for the cell to make proteins, and so there's DNA in every single cell. So DNA is a huge molecule. In fact, it's the biggest of the four macromolecules. DNA is huge, probably the biggest molecule known to man. That we know of. DNA is the longest and largest molecule of atoms connected to each other. Crazy cool structure and everything. So let's get right in now. Let me switch over to my PowerPoint. We're looking at Biochem Nucleic Acid chapter, which is 17 in this book. I know you don't have it, so I uploaded it to Google Classroom for you. Okay. Basically, our first distinction that we want to make is between nucleosides and nucleotides. So DNA, nucleic acids are made up of nucleotides. That's the word that's commonly thrown around. And you guys probably remember that from freshman biology. Hopefully a lot of this stuff rings a bell to you. If so, you'll do fine in this chapter. Nucleotides make up DNA. In fact, remember, DNA is a polymer made up of monomers, in which the monomers are nucleotides. So a chain of monomers make a polymer. A chain of nucleotides make DNA. But nucleosides, you may not have been familiar with. We don't talk about nucleosides very much, but they're important, and they're part of a nucleotide. So basically you have, first of all, two types of nucleic acid. You have DNA, which stands for deoxyribonucleic acid, and you have RNA, which stands for ribonucleic acid. Only a few minor differences, but end up being major differences. First of all, DNA, if you want to circle a word, here's the word to circle, stores your genetic information. So all of your genetic information, all of the genes that are on your chromosomes that make you who you are stored in your DNA. DNA is a storage molecule for an organism. And that allows you also to take your DNA and transfer it or transmit it to another organism, so that when you have a child, it has half of your DNA and half of your wife's DNA or husband's DNA. Now, ribonucleic acid is what translates that DNA. So something's got to translate or read that stored information, and it's the RNA, it's the ribonucleic acid that translates. That's the word to circle in that sentence. It translates the genetic information that's contained in the DNA. into proteins needed for cellular functions. Remember, DNA ultimately codes for making proteins, and it's the RNA that reads the DNA to do that. The nucleotide monomers that make up DNA look like this. I have this on the back wall of my classroom in a vinyl decal up in the corner. I'm sure you guys have seen it before. I'm just kind of fascinated with this molecule because this molecule is essentially like what is used in the DNA to code for proteins to make us who we are. So literally who we are is determined by a code in our DNA, and the code is written using this character. This nucleotide is like a character in the coding system. Computers use a coding system like zeros and ones, and DNA so it's very binary. It just has zeros and ones, open circuit or closed circuit. But your DNA, you can store information in it with a quaternary code because there's actually 4 different nucleotides, A, C, T, and G. And we'll talk about those in just a minute. But basically, they all look very, very similar. Only a minor difference in the nitrogen-containing base. But your nucleotide is pretty much the building block of life. Your building block of life is this nucleotide. And so I just think it's a very special molecule. Okay, DNA molecules contain several million of these nucleotides joined together, while RNA is a little bit shorter, and they'll only have perhaps a few thousand nucleotides. That's still a lot. But it's nothing compared to several million. DNA is, again, found in your chromosomes inside the nucleus of your cell, and each chromosome is going to have a different type of DNA. So chromosome number 1 might code for a certain amount of proteins, and your chromosome number 23 will code for other proteins. In fact, that 23rd chromosome is your sex chromosome. It's either X or Y. And if you have two X's, you're female. If you have an X and a Y, then you're a male because you got the Y chromosome. But humans have 23 chromosomes. All organisms are different. Different number of chromosomes, but we have 23 chromosomes. Now, of those chromosomes that code for proteins, we have a version from mom and a version from dad. So if you remember from biology, those are homologous chromosomes. They code for the same thing. They code for the same traits, but they're different because one's dad's version and one's mom's version. They're essentially recipes to make proteins, but dad has a different recipe than mom. So we all have 23 pairs. So essentially we have 46 chromosomes, but the way they're numbered is 1 through 23. And then on each chromosome, you have a section of DNA that has a starting point and a stopping point of which the cell will read that is going to end up translating to be one protein, and that is called a gene. A gene is just a portion of DNA molecule that's eventually going to be responsible for the synthesis of a single protein. All right. In a nutshell, that gene is essentially the information to make the protein. We begin our study of nucleic acids with a look at the structure and formation of this nucleotide. So we're going to look at how do we make this nucleotide, and that's where that word nucleoside is going to come into play. So nucleosides are just simply joining the sugar and the base. Let me scroll back up here again. The nucleotide is made up of these 3 components. I'm sorry I skipped this. A monosaccharide, Remember, of the monosaccharides, we have all kinds, but in this case, it's usually going to be either ribose or deoxyribose. And it also has a phosphate, which is just PO4, or phosphorus with 4 oxygens. And it also has a nitrogenous-containing base, a nitrogen-containing base, which we call a nitrogenous base. So it's almost like, it's not an amino acid or anything like that, but it's just there's a whole lot of nitrogen in it. And so we have the nitrogen-containing base. This is what's different in each nucleotide. Every nucleotide always has the sugar and the phosphate, in fact, you'll see later on, that's how you make the backbone of the nucleic acid, is by just connecting sugars with phosphates to sugar to phosphate, sugar, phosphate, sugar. And you get the backbone of the DNA or RNA, but the nitrogenous bases are what differ, and those are like the rungs of the ladder inside the molecule. and one nitrogenous base will always pair with another one to make the DNA molecule. Okay, so, but again, you have these three components: nitrogenous base, the sugar, and the phosphate. Well, a nucleoside is just simply joining the sugar and the phosphate. Oh no, I'm sorry. The sugar and the nitrogenous base. Forget about the phosphate for now. If you join just the sugar and a nitrogenous base, you'll get a nucleoside. And so let's first look at the sugars. For RNA, you guessed it, the sugar is ribose. For DNA, you guessed it, the sugar is deoxyribose. So they're basically the same pentose. They're both aldopentoses, which means pentose means it's a 5-carbon sugar. Aldo means it has the aldehyde group on it, right? So they're both aldopentoses. Everything is identical except for right here on the second carbon. For ribose, you have a hydroxyl group. But on deoxyribose, you don't. That's why it's called deoxy. You don't have that hydroxyl group. There's no OH group at the second carbon. This is real important too, guys, by the way. The carbons that are in this sugar are numbered, and they're numbered counterclockwise like this. 1, 2, 3, 4, 5. The 5th carbon's hanging off of the pentagon. So at carbon 2, you either have a hydroxy or don't. That's what makes the difference between ribose and deoxyribose, or D2-deoxyribose to be specific. So DNA has deoxyribose, RNA has ribose. Okay? Deoxy really means without oxygen, but okay. Now the nitrogenous bases, you essentially have 4 of them, but RNA replaces thymine with uracil. We'll get into that in a minute, but so there's really actually 5 nitrogenous bases total that are used in, if you want to just talk in general about nucleic acid, whether it's DNA or RNA, there's 5 nitrogenous bases, and we can classify these 5 bases into a class called pyrimidines or purines, and basically that means it depends on what the parent compound was. So if you start out with pyrimidine and you add a few things to it, you'll have these different nitrogenous bases. Just a 1-ring structure. Pyrimidine, it's a 1-ring hexagon structure with nitrogens in it. And if you add, let's say, a ketone there, an amino group, you get cytosine. If you add a ketone here and here, and a hydrogen here and here, you end up with uracil. So you get the point? Purines are adenine and guanine, and they start out like this. They start out with like purine, and then if you add like an amino group here, you'll be adenine. If you add a ketone there and an amino group here, then you'll be guanine. So very similar structures, right? Your purines look very similar. Adenine looks real similar to guanine. As well, your pyrimidines all look pretty similar. Cytosine, uracil, and thymine all look pretty similar in the fact that they're just 1 hexagon. And the purines stand out because they're basically a hexagon with a pentagon on it as well. So you have basically two ring structures. I haven't really found a good way to memorize this. I don't know how you guys can memorize that, but if you can come up with a mnemonic, please share with me how you have learned to tell, because I cannot think of something. I've tried and tried and tried to remember that the 2-ring structures are purines and just the 1-ring structures are pyrimidines, and I can't do it. I just can't seem to memorize it. And these are cool. The nitrogenous bases are, remember, what differ. So when you're writing a computer code, you can put 10001000111001, and that'll code for the computer to make a picture of a dog or something like that, or to open up a program. Your next computer input could be 00001001, and so on, and that code for something different. Well, it's the zeros and ones that are changing that are making the computer code do different stuff. Well, these are like the ones and zeros in the computer code. You have these nitrogenous bases that will differ in their sequence on the nucleic acid. And so these are very special. These are kind of like what end up dictating what proteins you're going to get, because the sugars and phosphates don't really change. Okay, so again, we're looking at nucleosides, and a nucleoside is formed by joining a carbon, sugar, carbohydrate sugar, and a nitrogenous base. And the way that happens is right here. You take the one carbon, remember these are numbered, you take the one carbon of the sugar and you combine it with this nitrogen at the bottom here, and look, you're going to lose this H and you're going to lose this OH. What are you going to make? Water. So it's a dehydration reaction, really. So I'm surprised they don't have over on the product side H2O. That's their fault for doing that. Same for this assembly here. If you take D2-deoxyribose and join it with adenine, you're going to get deoxyadenosine. Okay, this is a nucleoside. This up here is a nucleoside. We call it cytidine, and it's a ribonucleoside. Down here, this is a deoxyribonucleoside. Okay, so let's analyze that again. We have a ribose up here coming together with cytosine. And so all you have to do to name these is replace, let's see, where do they talk about that? Oh, it's down lower. For now, basically a nucleoside is just called an N-glycoside. And we use these little asterisks to refer to the primes for the carbons. So here's an N-glycoside, here's an N-glycoside. Both of these are N-glycosides. We call them nucleosides, but their specific name has to do with what sugar and what nitrogenous base you're putting together. And so for naming them, basically here's how you do it. To name a nucleoside derived from a pyrimidine base, you have to use the suffix -ine. So pyrimidine, you use the suffix -ine, I-D-I-N-E. Okay. So in other words, if you had cytosine as your nitrogenous base, it becomes cytidine. For a nucleoside that comes from a purine base, remember purine, it doesn't have the -idine, so you just use the suffix -acene. I know it's kind of weird, but where'd that S come from? But you just have to memorize that. So if you had adenine as your nitrogenous base, it would be adenosine. So the suffixes will show up as either adene or acene, and it depends on whether you're starting out as a pyrimidine or a purine. That's why it's pretty important to be able to look at this and immediately go, oh, that is a pyrimidine, and this is a purine. Because if it's a pyrimidine, you have to add adene as a suffix. If it's a purine, you add acene, O-S-I-N-E, as a suffix. And finally, if you have a deoxy sugar, if you have a deoxyribose, like you see here, remember there's no OH group here. Up here you do have one, that's ribose, but this is deoxyribose. If you have that, you have to add that prefix deoxy in front too. So for deoxyribonucleosides, you add that prefix deoxy. So here's a sample problem. It says identify the base and the monosaccharide used to form the following nucleoside, and then name it. Okay, so the base, you just have to look at it, and it's kind of like a Where's Waldo game. Like, which one is it up here? Which one does it match up with? Well, you can see that it matches up with thymine, because of the two ketone groups and the methyl group. You have the two ketone groups and the methyl group there. It's a one-ring structure. That's thymine. I don't have the OH group there on carbon 2 of my monosaccharide, so that would be deoxyribose. So it would be, if you wanted to name it, remember you have to use the deoxy prefix. It would be deoxy, and instead of thymine, it becomes thymacine or thymidine. You have to remember, is this a purine or a pyrimidine? And again, I haven't memorized it yet. Well, I kind of have, but I don't have a mnemonic to teach you. but I do know that this would be a pyrimidine, and so that becomes thymidine. So deoxythymidine. Deoxythymidine. Am I right? Yep, deoxythymidine. There you go. So here's 2 for you to try and do on your own. Also, they give you the opportunity to draw one, so I'll be looking for that in your homework so that you can you draw guanosine and also classify it as either a ribonucleoside or a deoxyribonucleoside. Okay, nucleotides, what are they? Well, they're basically just nucleosides, but now with a phosphate. And you always join the phosphate to the 5' carbon. Remember, the 5' carbon is the one that's like hanging off of the pentagon. So the way we number the carbons, we start here with 1 and then we go counterclockwise, or I'm sorry, clockwise. You go 1, 2, 3, 4, and then the 5th carbon is hanging off. And what you do is you take away this OH group. Wait. You take away this OH group. Yeah, I don't know why they're not going into the synthesis of this reaction here. I want to say this is a dehydration reaction as well, but where's the hydrogen come from? So it might need a hydrogen catalyst, an acidic catalyst. I don't know. But whatever. The point is that hydroxy does go away. And you end up forming this molecule here, or this bond here. The oxygen here on the phosphate gets bonded to this carbon because this OH is going to leave. And so this carbon is bonded to this oxygen. That's called a phosphodiester bond, actually. Well, not quite. Point is actually here that nucleotides are formed once you join the phosphates, and they always join here at the 5' carbon, okay? And then you have your nucleotides. Now again, if you're starting out as a ribose sugar like this, you would have a ribonucleotide. If you're starting out with deoxyriboses down here for your sugar, then you would have deoxyribonucleotides. And you also want to specify the 5' carbon is where the phosphate is. So like here you see the nucleoside is cytidine, which that comes from the fact that we have ribose and cytosine. You add the phosphate and now you have cytidine 5' monophosphate. It's always going to be 5'. In this case, we only have one phosphate, so it's monophosphate. Down here, we have the nucleoside is simply a deoxyribose sugar, and we have adenine, so the nucleoside is deoxyadenosine. Okay, so it's deoxyadenosine. Now, once you add the phosphate to the 5' carbon, it's going to be deoxyadenosine 5' monophosphate. And so that's kind of wordy, so we have a little shorthand abbreviation way of writing those with little 3-letter abbreviations. And just so you know, they always end in monophosphate, so it's MP, at least for these nucleotides. But they could have a lowercase d if it's a deoxy, so they could be 4 characters long. There is a nice little table here that organizes it for us, so check it out. For DNA, if you use the following nitrogenous bases with the following, well, you're always going to be coming together with deoxyribose if it's DNA, right? So you're always going to end up getting these nucleosides: deoxyadenosine, deoxyguanosine, deoxycytidine, deoxythymidine. Then the nucleotides you would get would be these, again, real wordy nucleosides added to the 5' monophosphate. Basically, you can just use these abbreviations here. Lowercase d if you have to add the deoxy. So all the DNAs will have that lowercase d there because you're dealing with deoxyribose. And then they'll always have the MP for nucleotides, because nucleotides only have one phosphate, so it's all monophosphate. Basically, the A, G, C, or T will come from the fact of whether you're using A, G, C, or T, adenine, guanine, cytosine, or thymine. Now for RNA, remember we don't use thymine. You'll never see thymine in RNA. You'll only see uracil instead. So uracil replaces thymine. And so if you want to go through the whole naming system again, you remember that we don't add deoxy. And so we just simply say the name, but we use the suffix acene if it's a purine, and we add the suffix idine if it's a pyrimidine. And then we always have the 5' monophosphate. And so here you go. UMP, CMP, GMP, or AMP. Now, does that mean that can we not have 2 phosphates, or can we not have 3 phosphates? Sure, we can actually, and this is what's really interesting. We've talked a lot before in biology about ATP, and how ATP is like the molecule that is like a charged battery because of the 3 phosphate groups here. And then if you knock off one of the phosphates, you have ADP, adenosine diphosphate. And so this would be like a dead battery. Well, if you put a bond there, if you put another phosphate right here, you'll have ATP again. And so charging and discharging this battery is simply the result of adding or taking away this 3rd phosphate. Isn't that cool? I didn't know this for a long time, that the ATP molecule is essentially like a nucleotide in our DNA. What a coincidence, right? What a coincidence. It's not really a coincidence when it comes to the way God designed us. He chose molecules for a specific reason, and the way they interact with us is all intentional, I believe. So, and if you study further biochemistry, you'll see how ATP is related to DNA and how it interacts with DNA. Okay, so here's a sample problem. Identify the base and the monosaccharide in the following nucleotide. So we have a nucleotide here, and they want us to answer what the base is, the nitrogenous base, and they want us to answer what the monosaccharide is, and they want us to name the nucleotide and give its 3 or 4 letter abbreviation. So we got a lot going on here, but it's all building on itself. Like if I want to name this thing, I got to know what kind of sugar it is. Is this is this ribose or deoxyribose? It's ribose, right? Because it's not missing the hydroxy. So this is ribose. What is this? Well, I have to scroll back up here and look, and it looks like that is guanine, because it has the NH2 poking out here to the right with the ketone up there on top. And so if we look at it again, it's got the ketone up top and the NH2 poking out on the right. So that is guanine. So it's deoxyguanidine or guanosine. Well, it's a purine, so it's acene. So guanosine. So deoxyguanosine. Actually, it's deoxyguanosine 5' monophosphate. So that would be lowercase d GMP. Did I get it right? No, it's not deoxy. Why did I say deoxy? It has the hydroxyl group, so it's just GMP. Guanosine 5' monophosphate. Okay, so you have some to do of your own now. So finish those. They give you plenty of work to do here, which is good. I want to get you guys working right away so you're familiar with this. The next lecture will be on nucleic acids, which is when we'll be actually joining these nucleotides together to form a long polymer of nucleic acid. All right, thanks for watching.